Welcome to Science Frontiers, a space for discussions in the frontier areas of science and technology. I am a host, Binay Panda. As students of biology, we have studied the definition of a gene and genome. And we know that the majority of the regions in the genome do not code for a particular protein. In fact, talking about human genome, depending on the study that you are referring to, only less than 5% of the entire genome codes for any protein. And in fact, a large majority of them are still, we do not know the function for. Now, apart from the protein coding genes, there are a lot of regions in the genome that code for RNA. However, taking together for both the protein and the RNA coding genes, there are a vast regions of the genome that do not code for anything. And for the lack of a better term, as you will understand from today's episode, let us call that as non-expressing part of the genome. In fact, in 1972, after studying the DNA of a variety of species, Susumu Ono, a scientist at the City of Hope National Medical Center in US, put forward a possible explanation. And he wrote in the Brookhaven Symposium in Biology, proposing that these millions of silent DNA sequences represented genes that had died off like dinosaurs during the course of human evolution. He said, and I quote, "The earth is strewn with fossil remains of extinct species. Is it a wonder then that our genome too is filled with remains of extinct genes?" However, Ono's paper is perhaps best remembered today for another reason. He titled his article, "So much junk DNA in our genome." And in fact, this term junk DNA stuck out, and thus born a field where people started studying the actual structure and function of DNA, which is very famously known as junk DNA. Now, today's episode, we'll talk more about what we call a junk DNA and why the junk is, after all, not so junk. According to most scientists, the answer remains still unknown. A growing body of evidence has begun to accumulate that suggests the junk DNA are, in fact, regulatory in function. We shall explore this topic with a new name, and you'll understand soon why we are calling this as the dark genome, with today's guest, Pawan Dhar. Pawan is a professor in the School of Biotechnology, Jawaharlal Nehru University, New Delhi. Pawan, welcome to the show, and thank you for joining us.
Thank you, Vinay. Thank you very much.
Now, Pawan, take us back to history and tell us why you call this as the dark matter of the genome, and why not the same as Ono told as junk DNA?
Thank you, Vinay. I think that's a wonderful introduction you have given. In fact, you made my job very easy because the term junk that Ono was referring to for so many decades, people have been following that religiously. Talks about sequences that eventually turned out to be RNA coding. So he thought that probably they are not doing anything, and a lot of scientists at that time thought in a similar way. But later on, people found that what was the junk, it was actually RNA coding. Now, the term dark matter differs from the term junk in one very important aspect, that is, there are three types of sequences in the genome if you look at from the functional point of view. One that encodes a protein. Second, that encode RNA, which was previously referred to as junk. But the third, that encode neither RNA nor protein. So even with the best annotation efforts, people have not been able to find any reason for their existence because they do not transcribe, they don't express. This is the section that we call as the dark matter of genome.
That's very good, Pawan. Now tell us something about the first experiment that you did with some key technical details that you may want to share with our viewers, how you tried doing or converting some of the non-expressing part of the DNA into functional proteins.
Thank you, Vinay. Our first experimental effort in this direction was back in 2008. And then we were trying to brainstorm in one of the lab meetings as to what could be the possible function of such sequences that nature has never looked at from the expression point of view. And logically, therefore, if you want to make a functional protein from non-expressing sequences, these are naturally non-expressing sequences. If you want to make functional protein out of it, you've got to make a gene out of it in the first place. So you have to convert, let's say, an intergenic sequence into a functional gene. Now, theoretically, it's very straightforward, because if you go by the textbook definition of what a gene looks like, so you have a promoter sequence, you have a ribosome binding site at the level of transcription and translation, you have the start codon, stop codon. So theoretically, if you have a promoter sequence, start and stop, you can convert an intergenic sequence. Well, this is what we were using at that time. In the first paper that you're referring to. So we could convert it into a functional gene. But if you play at the genome sequence level, it's a lot of work, because you have to ensure that your sequences, that the signals that you're installing are in the right order. There is no mutation, there's no gap. That would be a lot of work. So what we did is, we thought, let's make a vector that will supply the start and stop codon along with promoter. And we will put some other markers there and then express it. We were very fortunate that Invitrogen had released PBAT topo vector around the same time. And this PBAT topo was a sort of a divine gift for us, because it provided not only the start and stop codon, it also provided the promoter sequence and ribosome binding site. And on the top of that, there was also a his tag. And now you know that when you express a sequence into functional protein, you want to know, is this your protein among thousands of other proteins that are found in the cell? And there are anti-his tag antibodies that you can use. And at the Western blot level, when you see the band, you know that, hey, this is my protein, as against a million other proteins that are swimming around. So this was the flow of the step. We started with bioinformatics. Our first intent was to ensure that these sequences have never been produced in any organism wherever the data has been collected from. So we converted intergenic sequences into protein sequence equivalents. And then we computationally match-made with all the other protein sequences that have been discovered so far from the non-redundant protein database of NCBI. And from there, whatever number of sequences whatever was filtered out, we were left with. We then asked another question, that are these conserved? And if these are conserved, we removed that also, because somebody might say that there's a reason for conservation. We didn't want to take that chance. So we took a random sample, different size of sequences, overlapping, non-overlapping, different directions. We took 10 sequences, and then we followed the story. We lost 4 sequences on the way, but in the first paper, we were able to express 6 intergenic sequences into functional proteins. And in one of them, we found a very clear function that it was killing the cell. And this was all done in.
With that, Pavan, I want to take you back to a philosophical question. As any student of molecular biology will tell you, as long as any DNA sequence bears signatures of expression, and what I mean by that, with the right beginning, right end, and the sequences for ribosomes to get hooked onto, it will produce protein. Now, in that context, tell us why nature decided not to produce any functional proteins from these non-expressing parts.
Yeah, that's a wonderful question again, Binay, because this has been asked many times in certain meetings. What we are claiming, and this needs to be understood by the audience in the proper context. When we say that we are making novel genes, novel RNA, novel proteins from the sequences that nature did not use, we are only referring to the data that has been collected so far. We are not talking about the entire infrastructure that nature has made across all the species and the entire biodiversity of the planet. We're not talking that because we do not know. We have hardly scratched the surface. So only with the reference to the data that is available currently in the public domain, we are referring to that data. Now, obviously, we know that nature, let's say from E. coli organisms where we made these new genes and new proteins. Well, nature had some reasons not to produce this raw material because E. coli is sufficient to carry its transactions. But it is just that we want to highlight the fact that we're not doing something against the nature, because these sequences are made by nature. We're just trying to find the purpose by expressing, which was designed by nature. It was not designed by our lab. But there are publications that indicate, there are many such publications that indicate that intergenic sequences over millions of years have become the protein coding genes in humans, Drosophila, E. coli, worm, and so on. There are publications that indicate that. So nature has a way to convert a sequence that was never expressing into a certain functional resource. But it takes millions of years. And we do this in the lab in 6 months. That's the only difference.
Now let us go back to the observation that you mentioned. And that observation is you found certain sequences, the non-expressing part of the DNA, toxic to the cells. Now, going back to the E. coli, what made you think that most of the non-expressing parts are actually not going to be toxic? And for that matter, apart from E. coli, in any other organisms, what made you think that you will not get toxicity or the phenotype which is less desirable that you wanted to?
Great, Vinay. The sequences that we considered initially, we collected this in a set of 10 sequences in E. coli because that's the organism we were using in the lab. And I must say that we were very lucky to get this phenotype. If we wouldn't have got the phenotype, out of 6 cell lines, only one of them gave us the growth inhibitory effect. If we wouldn't have got this phenotype, probably we may have thought of moving on and doing something else. But because we were very fortunate, the second question was, will we be fortunate again, or is this a random observation, as you rightly mentioned? So to ensure that next time it's not the luck, but a certain narrowed down sample size that gives us results, what we have done is we have moved the entire project from experiment to competition. So then we designed a database of the lab-made gene database, wherein we sample every intergenic sequence from E. coli, and we also took many sequences from yeast and Drosophila and humans. With this assumption that whatever was non-expressing, if we hypothetically make genes out of these sequences, we make RNA and protein out of these sequences, what would be the structure of the protein? What would be the function of the protein? What would be the interaction pattern of the protein? What would be the cellular address of the protein? Will it be stable or will it be unstable? So when we did all of that, it took us several years and a lot of PhD students to construct this database. Now the question is very simple. The question is, if we want to make, let's say, anti-cancer protein or an anti-cancer peptide, show me the topmost ones, the ones that are most likely to generate. So now, after the first experiment, which was published in 2009, then we moved on and we made anti-malarial peptides from yeast intergenic sequences against 3 different targets. And then we made an antimicrobial peptide from the intergenic sequence of Drosophila melanogaster. Subsequently, we made something against the breast cancer. And very recently, we have generated anti-Alzheimer peptides. They're showing very good results. I must say here that one of the crazy ideas that we had some time back, going a little beyond this work, was, can we make a functional tRNA encoded peptide? Because tRNA never expressed itself in the form of polypeptide. It was doing this kind of an outreach program for millions of years, ferrying amino acids from one place to the ribosome, and sort of helping the cell to make these polypeptides. But it itself never got translated because of many reasons. We don't get into that structural biology. So what we said is that could we make functional tRNA peptides? And if yes, what would be their structure, function, and can we generate such molecules? So we performed bioinformatics, found the right molecules after going through a lot of data. And finally, we made a tRNA encoded peptide that we call as trep. And what was amazing is that it gave us a very strong growth inhibitory effect on leishmania. And the IC50 value was close to 30 nanomolar. And that was really striking because we realized that this is an untapped gold mine, and that's exactly what you're referring to.
This is very fascinating, Pawan. Here we are repurposing biological molecules like the one you mentioned, tRNA, that we know is not designed to make protein. And when you repurpose tRNA to make novel polypeptide that is therapeutic, it is extremely interesting. In fact, I want to ask you a question that is related to evolution. And this is very timely for our viewers if I remind that one of the famous evolutionary biologists, the American-Russian, and had even alive today, would have been called American-Ukrainian, Theodore Dobzhansky, who said, nothing in biology makes sense except in the light of evolution. And what I want to relate the question back to evolution is it far-fetched to think that these sequences which are non-expressing today, for example, what you mentioned about tRNA, once upon a time were expressing and actually were producing polypeptides?
That's a brilliant thought, Binay. We have been thinking about this. How was the pre-ribosome world like? Did it have a machinery, a pre-ribosomal machinery to translate, sequences into polypeptides? Obviously, we do not have the answer. But the dark genome content that we are currently investigating, it comprises of like tRNA sequence, it comprises of intergenic sequence, it comprises of antisense proteins. And we have made reverse protein. That is, when you're reversing the coding sequence and you make another protein, we have made some functional molecules from there. We have made molecules from we are now in the process of making molecules from introns and repetitive sequences. Why are we doing that is only because we want to go back into the evolutionary domain and answer this question that why during evolution these molecules were not made, or have we looked far enough? Are there surprises around the corner that we're not aware of? This is a new biology, and it is not contradicting the standard biology, but it is just building on the top of what we already know, because there are no exceptions in nature. There are only lovely decisions that we need to uncover.
My last question, Pawan. Now that you have mentioned about expressing both non-therapeutic and therapeutic peptides from the non-expressing parts of the genomes, what are your future plans?
Great. Vinay, you see, as you already mentioned in your opening statement, you see a minority of the genome, let's say in humans, close to 1.5%. It depends on people whom you talk to. It could be lesser than that. Close to that is protein coding. And now we know that a large fraction of human genome is RNA coding. A huge content of the genome does not seem to be expressing. Now imagine if you convert 98.5% into functional polypeptides. If you convert the entire RNA coding and non-expressing genome of E. coli and Drosophila or other organisms into functional polypeptides, you'll have a huge inventory. I mean, we don't have the infrastructure to create so many proteins. So this is functional genomics 2.0, as we call it. Focusing on those sequences in future, we are going to focus on those sequences which have been ignored in the standard biology, context. So definitely, we are in the process of making therapeutic molecules, but we are also in the process of making enzymes from this non-expressing genome. We are in the process of making transcription factors and signaling molecules. And I must quickly mention this. This is a work in progress, but we have got some indicators that we are able to make a functional restriction enzyme from an intergenic sequence of E. coli.
I'm afraid that's all the time that we have for today. With that fascinating note, we would like to end today's discussion, and thanks, Pawan, for giving time and being present in the discussion. Thank you once again, Pawan.
Thank you very much for this wonderful talk.